Hello, my name is Emil Bernal, and today I'm going to explain and I'm going to present the inhibitory drug for cancer cells, tamoxifen. Mechanisms of action of tamoxifen. Tamoxifen is a non-steroidal drug used as selective estrogen receptor modulator called SERM and antagonist to estrogen action. Breast cancer development and progression are influenced by steroid hormones, particularly estrogen, via their interaction with specific target receptors, in this case estrogen receptors. Tamoxifen functions as an antimetabolite. It inhibits DNA synthesis as competitive inhibitor that binds enzymes in place of estrogen. Tamoxifen can be agonistic in other tissues like uterus, the bone, and the heart. So we want to see these side effects in the next slides and how they are produced. So in this picture on the right, we have the receptors, the estrogen receptor, whenever they are bind to serum, and some RK that binds growth factors, and they produce cellular signals. These cellular signals, they phosphorylate some core regulators that forms a complex with serum and the estrogen receptors, in which they go into the cell and into the promoter region, in which they generate the gene transcription for activation or repression of the biological effects. So some of the failures of apoptosis happen when the p53 gene is mutated or degraded by chemicals, or the E7 bind to the RB proteins and phosphorylate them and has the restriction point in which the cell proceeds through the cell cycle. Like for example, we have here a picture on the right in which this virus produce these two proteins, the E6 and the E7. So the E6 protein, it stimulates ubiquin to bind P53. P53 is necessary for the cell to go through the apoptosis, and whenever it is degraded by ubiquin, no longer the cells go through apoptosis. The E7 proteins binds to the RB protein. The RB protein is necessary to test the cell to be in the right conditions to go through the G1 to the S phase. Whenever it is phosphorylated, this protein no longer tests the cell to go through the restriction point, in which the cell just go from the G1 to the S phase. And tamoxifen works in this way. They produce a lot of CD cyclins because of the growth factors that they produce, and these cyclins phosphorylates these RB proteins in which the effect is done. So tamoxifen action pathway in cancer cells and normal cells. So two major isoforms of estrogen receptor we have the ERA and ERB that promote physiological processes, including the development of female reproductive system, the maintenance of the bone mass, and the protection of the cardiovascular tissue and the central nervous system. And this protein has different domains called activation domains. This, for example, we have the ERA, part of the estrogen receptor protein, in which we have AF1, and we have AF2, which is the ligand binding. We have the DNA binding in C, and D, we have the promoter binding. So tamoxifen binds to the ERA, resulting in the recruitment of co-repressors, NCORA and SMRT, to the ERE of the target genes, causing repression of the ERA signaling in breast cancer cells. As we can see right here, the SERMs, which is the tamoxifen, binds to the ER receptors, and it recruits the co-receptors, the co-repressors, and then gene transcription is inhibited. So in normal cells, the co-activators or co-repressors, the growth factors, protein kinases like CDKs, regulate the amount of material inside the cell to specific levels. And like for example right here, we have estradiol that binds to the estradiol receptors, but some aromatase inhibitors can destroy these hormones in which they cannot bind the ER enzyme, in which they cannot generate any gene transcription in normal cells. So cells can shut down signaling events, reducing the amount of free ligand, reducing the total amount of the signal, or by reducing the sensitivity of the receptor or the amount of receptor in the cell. So in normal cells, they shut down these enzymes inside the cell in order for them to not produce too many signals. And in this way, they inhibit the gene transcription. So the major effects of tamoxifen and relationship to its pathway. So we know that the serious effects of tamoxifen are uterine cancer, blood clot, strokes, and cataracts. Other side effects of tamoxifen include menopause-like symptoms such as hot flashes and vaginal discharge or bleeding. So it happens by the recruitment of NCOIA, which is a highly expressed coactivator in the uterus. It's responsible for the agonistic effects of tamoxifen by binding to the ERA complex on promoters of tamoxifen sensitive genes. So another actions of the cell that have is like whenever they produce a lot of growth factors, like which activates the pathway, like this one, the IGFIR, in which they produce a lot of P13K kinases, and these P13K kinases, they promote a cascade of phosphorylation, like how we see it right here is phosphorylase AKT and then mTOR, and this mTOR phosphorylates the estrogen receptors, in which they attract coactivators and transcription factors and affect and produce the gene transcription. So even if no hormone is bind to the estrogen receptors. So another thing, like whenever growth factors bind to the ER or hormones bind to the ER, they send signals for cyclin to bind to phosphorylate cyclin, and this phosphorylation creates the CDK4/6 to bind cyclin, and this complex phosphorylates the RB protein. The RB protein, whenever it's together with the E2 factor, they don't produce any gene expression. But whenever the E2F factor is free, they produce cell cycle progression, in which the cancer cells are activated and proliferated. So a related topic question is the human estrogen receptor ER is a ligand-inducible transcription factor that contains two transcriptional coactivator functions, one located at the N-terminal region of the protein in the AF1 region, and the second is in the C-terminal region, known as the AF2. It is known that some breast cancer cells inhibits tamoxifen antagonism, and instead tamoxifen promotes region or promotes breast cancer proliferation. So my question is how tamoxifen becomes agonistic in cancer cells proliferation through the AF1 transcriptional activation terminal. Alright, thank you for watching this video. Bye.